So we're moving on now to look at our next theme, accelerating novel therapies. So it's a great pleasure to welcome the co-lead of this theme, Professor Manju Kurian, who's making her way down now. Manju is a professor of neurogenetics at UCL's Great Ormond Street Institute of Child Health, and she's an NIHR research professor. She's also a consultant pediatric neurologist at GOSH. Thank you very much, Manju.
Lovely. So my name is Manju. It's a great pleasure to be here and a great privilege to be co-lead of the Accelerating Novel Therapies theme together with my co-lead Darren, our deputies Hyan and Harry, our junior representatives Wendy, Will and Mansa, and of course we'd be nowhere without our research manager Azizat. So just to kind of put it in context, I think something that we think about all the time working at Great Ormond Street with lots of rare disease cohorts, children that have very serious complex conditions, is that actually 75% of rare disease affects children, and frighteningly and devastatingly, 1/3 of these children die before their 5th birthday. And so our Accelerating Novel Therapies theme, the ANT theme, really has a major focus on developing better medicines for children that are tailored to their diseases. And these are often for children that have rare, life-limiting diseases with the aim of a cure, better quality of life, or a higher chance of survival into adulthood. And to do that within our Accelerating Novel Therapies theme, we aim to do this by really crossing that translational arc. And what I mean by that is really going from the bench or the laboratory kind of basis, or the campus, the university campus, all the way to the patient bedside or the clinic. So that's a big arc from undertaking basic science all the way to patients and trials and then licensed treatments within the NHS. And our theme really does focus on that arc. And how do we do that? Well, we're aiming to develop better state-of-the-art laboratory models of rare disease, developing biomarkers or tests that can tell us how effective treatments are and also to track disease over time. We're developing some very new and novel genetic therapies and also undertaking some very cutting-edge first-in-human and first-in-child clinical trials at Great Ormond Street. So to really start from the bench, really one of the main things that we struggle with is having the right models to allow us to investigate disease and develop new treatments. And you've heard a little bit about organoids already, and one of our theme's main focus is to develop these organoids that represent human tissue, and we use those to understand disease and develop new treatments. And for example, what you're seeing here, I think it's a very pretty picture that looks like a flower, but it's actually part of the brain called the midbrain, and it's really useful for trying to understand diseases in children that have movement problems. So you can analyze this kind of organoid to find out how disease happens, and you can also use this organoid to treat with gene therapy, with small molecules, with drugs, to see whether you can correct the problems that the genes cause. So that's one aspect of our work, and we make models in the brain, in the kidney, in the lung, all of these to help us understand disease and develop new treatments. Another thing that we really think is lacking and that we've really tried to develop is a technique called CRISPR. And what CRISPR is, it's basically a tool that allows you to change the genetic code. So we all carry genetic code, and that's like a book. And you may want to change a sentence or a letter. And this is Shalini Reddy, who heads up our CRISPR service. And that allows us to edit genes to allow us to make genetic mutations that are in our patients or correct genetic mutations. And we use that to improve diagnosis in our patients. We use that to understand disease mechanisms. We use that to screen kind of our lab models to be able to understand what pathways are involved. And most importantly, we can use it as a new tool in the future, as a therapy. So you can imagine if a child has a genetic mutation or a genetic spelling mistake, the ultimate treatment would be to see if you could correct that spelling mistake within a time frame to allow that child to live a normal life. As well as this, within the laboratory part of our theme, we work on developing biomarkers, and in this BRC, we've developed several new tests that are being used in industry-led clinical trials and also academic trials within the NHS. These markers can be used to monitor and track disease for lung disorders, endocrine disorders related to hormones, muscle diseases, neurological degenerative diseases, and this will tell us about how the disease tracks over time and whether it can also help us determine whether a therapy that we're giving has an impact on patients. And as a result of all this kind of success, the leaders in this area are a key partner in a national initiative looking at how lipid or fat markers, fat biomarkers, can tell us about disease. And this is all culminated in this group involving Wendy, who will speak in a minute, and Kevin Mills, spinning out a new company called Guildford Street Laboratories, which aims to accelerate that development of these biomarkers and rapidly bring them into the clinic. We're also developing new genetic therapies, and these are called RNA genetic therapies. This is work led by Haiyan Zhu in our theme. And I think one of the most successful things she's done is introduce a really broad international network. So she's based in London, but literally she has collaborations everywhere. And all these collaborations and network have really accelerated the development of these novel RNA therapies. So much so that hopefully very soon at Great Ormond Street, with work led by Professor Gisson, we'll be able to bring these tailored treatments, what we call an N of 1 treatment, where the genetic treatment is for a specific patient to each of our patients in Great Ormond Street Hospital. We're also doing a number of clinical trials that are really cutting edge, and here's a great example of work led by Professor Hargrave, who has evaluated the effect of a new cancer treatment for a form of brain cancer, which is seen to be effective. And we're now undertaking trials, including trials for children with what were previously considered to be life-limiting neurodegenerative conditions. So that's an overall view of our theme. In the next, looking ahead, what we'd like to do is really use those laboratory models I've talked about to test drugs and accelerate that development to patient trials and then to licensed treatments. I think we're very close to initiating this RNA genetic personalised therapy for our patients at GOSH. And it'll be really nice to see whether this one medicine, one patient treatments are successful. We want to really enhance national and global collaborations, because what we realise is that the only way to tackle rare disease is really by working with people globally. And I think very importantly, with all of these themes, we want to train the next generation of ant researchers. And with that, I'd really like to introduce Wendy and Francesca, who will be speaking on behalf of our theme. Thank you very much.
Good afternoon, everyone. So I'm just going to give you really just a brief overview of the story of Immunotadar, which is an innovative anti-drug antibody assay that we've developed. And I'm going to tell you the story of how we've gone really from bench to bedside here. So one of the challenges of treating patients with rare diseases is that the immune system can develop antibodies to the treatment. And this is particularly a problem when it comes to treatments such as enzyme replacement therapies and also gene therapies. Now, these antibodies can make the treatment ineffective and can also cause adverse reactions, particularly when it comes to gene therapy as well. Now, Fabry disease is an inborn error of metabolism that results from dysfunction of the enzyme alpha-galactosidase. Now, there are 3 enzyme replacement therapies based on alpha-galactosidase that are on the market. And each one of these costs over £100,000 per patient per year. Now, 20% of those patients develop ADAs, or anti-drug antibodies. So that's a lot of treatment, a lot of expensive treatment that is not effective, and obviously costing the NHS a lot of money. So there needs to be a better way of assessing anti-drug antibodies, and the current tests that are available are actually only provided by the pharma companies themselves, which, as you can imagine, is not an ideal situation. So we've developed an independent ADA testing assay service. Now, Immunotarda, or immunotargeted antidrug antibody assay, was first conceived for SARS-CoV-2 back during the pandemic and has been adapted to measure ADAs, particularly for Fabry disease in this instance. Now, the platform is actually an absolute quantitative analytical platform using targeted mass spectrometry. Now, the principle of Immunotarda is quite similar in the first instance to a normal ELISA. You have your immobilized bait protein, which is your antigen, or your, in this case would be your ERT drug. And then you would add your patient sample in, incubate, and specific antibodies will then bind to your antigen, and then you would wash off anything that's non-binding or non-specific. Now, when it comes to an ELISA, you add a secondary antibody and you end up having a colorimetric reaction that you would measure using a plate reader. Now, with Immunostar, what we do here is we don't add any secondary antibody. We essentially just digest it. We break it up using proteolytic digestion, and that includes breaking up the whole immune complex that's bound to the ERT. That breaks down into peptides, and those peptides are then quantitatively and accurately measured using targeted mass spectrometry. Now, the benefits of this platform are that it provides absolute quantitation, not titers. It provides an internal control system, which you can't really get with ELISAs because you're actually measuring the bait protein as well, which you can then ratio and normalize. So that actually enables better quality control. It has a wide dynamic range because you're not amplifying up your signal, so there's no need for dilution of samples. There's no requirement for secondary antibodies, which I'm sure many of you are aware can have issues with batch-to-batch variation and also costly. And also, it's an independent platform that's not dominated by commercial integrated systems, which is what you have in a lot of clinical laboratories. But one of the USPs or unique selling points of Immunotarda is its multiplexing ability. So we can measure not just IgG, we can measure all the subgroups of IgG, 1 to 4. We can measure all the others, such as IgG-M, IgG-A, even IgG-E if it's there. What we can also measure are the proteins that bind to immunoglobulins, and that includes complement. So we can actually tell so much more from one injection or one reaction about the immune response. So we've now sort of developed that into a targeted assay, and we applied this to a cohort of samples that were provided by Professor Darren Hughes at the Royal Free, who treats Fabry patients. And the assay was adapted from the original SARS-CoV-2 platform to ERT by Dr. Tom Baldwin. Now, the key findings that we had from analysing the cohort of samples were that patients on ERT treatment have different ADA and complement profiles, which you wouldn't capture by a normal ELISA. And the main immunoglobulin that's responsible for ERT inhibition is IgG4, which corresponds with some previous published work. And this is in Fabry males. Now, we also were able to determine, or able to show, that you have less IgG4 binding to a less immunogenic form of ERT that has recently come out onto the market. And this has actually resulted now in patients being switched to this new treatment. We were also able to show that an adverse immune reaction was caused by an IgA2 complement activation in 2 patients who were receiving ERT. And again, that is something that you couldn't pick up by using conventional ELISAs. Now, Immunotarda has come a long way. As I mentioned before, its initial concept was during the pandemic, where we received some funding from the BRC to develop that against SARS-CoV-2. We then patented that with UCLB, that technology, and that enabled us to get tech transfer funding from the TRO in 2023 to develop the platform for Fabry ADAs. And then with that, we were able to license the patent and spin the technology out into Guildford Street Laboratories. And Guildford Street Laboratories are now developing the platform in the NHS clinical lab at the National Hospital, which we will now be able to offer out as a service for clinicians to order in the UK from the end of this year. Now, the brilliance of Immunotarda, or the benefits of this, is that we've designed this platform so that we can actually swap out that bait quite easily. So we can actually develop it also for other ERT drugs and any other biologics. But also, we've been developing this also for anti-AAV as well. So we also have some results for anti-AAV work that we hope to show at some point as well. And I'd just like to thank all of those that have been involved in this translational journey, particularly all those at ICH. And this is really a collaboration with Great Ormond Street Hospital and the Royal Free Hospital. and the clinical translation, which has also been done by Guildford Street Laboratories with a grant from Chiesi and also with the National Hospital.
Thanks, Wendy. Francesca.
So hi, everyone. I'm Francesca. I'm from the University of Liverpool and Alderhay, as you can see. So thank you very much for giving me the opportunity to present here today. And so I will talk a little bit about the research we do in the Institute in the Park in Alderhay. So we are based in Liverpool, and actually the Institute is home to different teams. And I'm part of the Experimental Arthritis Treatment Center for Children, so for short, EATC for children. And so in case you want to know more about what we do and who we are, you can just scan the QR code and you will be brought to our website. So despite what the name suggests, we're actually not only interested in rheumatic diseases, so we don't only study rheumatic diseases, but we are interested in inflammation in general. So we also had some projects on cystic fibrosis, for example, or on COVID-19. We do have different projects, but we have common objectives for all of them. So the main objectives for us are to find new biomarkers, to identify genetic and epigenetic signatures that could help us in the diagnosis of patients, and also I would like to identify new treatment targets. And then I just wrote a quote there from our website, and it's just to highlight the importance of collaboration, especially when working with very rare diseases. And this collaboration can really help in helping the well-being of our patients. So here is a list of all the projects we have in collaboration with GOSH BRC. I will just go very briefly through them. And then here on the right, I added a map of all the collaborations we have in the UK. So PASTIS is a study about juvenile onset systemic scleroderma, so an autoimmune disease that affects the connective tissue. And then we have Detective, that is about chronic non-bacterial osteomyelitis, so an autoinflammatory disease that causes the recurrent inflammation within the bones. And then we have Trojan and TLR7 variants projects that are about juvenile onset systemic lupus erythematosus, so another very rare autoimmune disease. So we do have different projects, but they tend to have all the same workflow. So we do start from patients, we collect the samples, and then we isolate DNA and RNA from them, and then we sequence them, and we also look at epigenetic marks, so more in detail, DNA methylation for now. And then we send all the data to our bioinformaticians, and then we obtain all the results back. And then what we do is to validate what they found in the lab, so to do some functional work. And then the aim is always to be able and go back to the patients to improve their care. So here is just an example of the TLR7 project, the TLR7 variants project about, and I mentioned before, about juvenile onset systemic lupus erythematosus. So what we did was to we identified this variant that was located in the last part of the gene, so in the 3' UTR region of the gene. And we noticed that this variant was associated with mucocutaneous involvement and also with older age at diagnosis and with higher anti-DNA antibodies. And what we noticed is that this variant made the transcript of TLR7 more stable, so it also resulted in a higher interferon score. And this is actually really important to understand both the diagnosis of patients and also to understand the disease and to also tailor treatments for the patients. So here's a very busy slide, I'm sorry. It's just to say that in the future we aim to continue following our objectives, so improve our diagnostic tools, to find new treatment targets, and then to also design a new tissue model to study the diseases we are interested in. And of course, we'll continue strengthening and building our network, both nationally and internationally. Thank you very much for your attention.
Thank you very much. If I could invite Manju to come back up with us, and Wendy as well. Thank you very much. I've just been told there are lots of seats available, so people can move down if you're stuck on the stairs at present. Please do move down. Thank you. So we have a series of questions, but actually I'm going to start. First of all, thank you, Jane. I'm going to start with the question for Wendy. Wendy, you mentioned, I think early on, that 20% of the patients have the ADAs. Do we know why 20% of people get them and 80% don't?
So the patients that tend to develop ADAs are the Fabry male patients. So Fabry disease is an X-linked disease. So if you result in one copy of the gene dysfunctional, then you're more susceptible. But these patients are the ones that tend to have non-mutations, so they have complete absence of the protein. So when you actually administer the protein with the ERT, it's completely foreign to their immune system, and that's why they build up an antibody response.
Thank you. Questions in the room at all? Are there online questions, David?
There certainly are. We'll stick with Wendy. I don't know whether you did any health economics work, but there's a question asking, do you know, what do you think the estimated cost difference would be for the immuno TADA compared to the current cost for ERT? I don't know whether you did that.
We haven't quite done the health economic cost just yet, but I mean, potentially it could save money. But the idea, I think, is that we could put patients on alternative treatment that's more effective, which are becoming more available.
There's a question at the back, please.
Question for Wendy, really exciting. Could the technology be adapted for drugs which are immunogenic that are themselves immunoglobulins? So they're not completely missing from, obviously in this case, the patient is not tolerant because they never had the gene in the first place. But we have many drugs and a problem with anti-drug antibodies that are immunoglobulins themselves, anti-TNFs and others.
Yes, it depends on the sequence of the drug and if it's shared very much with the humans. But if there's a specific protein, or sorry, peptide that can distinguish it, then potentially we could develop that, yeah.
Thank you very much. I'm going to ask Francesca. Obviously, you showed a huge network there of different areas you're working with. Sounds a slightly naive question, but what's the benefit for you individually in your projects in having such large collaborative networks?
So I presented all the projects for our team, but I'm not working like myself on all of them.
Generally.
Yes. Yeah, so I think it's really important because there are some things in common between different diseases. So it's always nice to know more. But also like this allows me to have to meet other people, other researchers, and share ideas. And this is actually really important also for our careers as young researchers.
Thank you. And it's wonderful for me to see that the Paediatric Excellence Initiative, which started in this term, has really grown almost a life of its own, I think, watching now, our partners in Alderhay and Sheffield and Birmingham as well, and how the network really is gradually growing and growing with more and more people making connections and productive uses in these interactions. I'm going to go back online. Thank you.
Yeah, there's a follow-up question there for Francesca. What would you say are the benefits of collaborating with GOSH?
So there are a lot of benefits actually. So one is the access to a lot of samples, of course, and then also to the knowledge that you have here at GOSH. So yeah, I think these are the two main that come to my mind right now. So it's really nice to be a partner for us.
Thank you very much.
Thank you.
Are there any more questions in the room? No. Any more online?
Yeah, I've got two for Manju. I'll try and wrap them up into one question. What are some of the challenges of translating research from bench to bedside, and in particular, are there some areas of which organoids are difficult to create?
They're really great questions, and whoever's asked them, they're the exact questions that as clinician scientists we spend our whole time spending time on. And I would say almost the preclinical work, in the laboratory, in cell models, in animal models, is done relatively easy for lots of diseases. But then bringing that to clinical trial is often very costly. It's often, there's lots of regulatory kind of processes to go through. I mean, that's where, I think work and research needs to go into kind of streamlining those processes. So it's not an easy process, but obviously we now are navigating it for a number of different conditions and a number of different pipelines with different kind of treatment kind of approaches. So genetic therapy, small molecules, etc. So I would say that the current bottleneck is really from the preclinical to attaining funding for rare diseases, to then navigating the regulatory framework that we need to navigate. And then clinical trials, again, they come with their own challenges. And then translating that to licensed treatments, I think, is another bottleneck. You know, understanding really at the laboratory stage what you need to please the regulators, so the MHRA, the FDA, EMA, right at the beginning, and to work on that so that when you get to that point of the end of a successful clinical trial, that there's not another bottleneck. So they're the things I think we need to think about as a community. And to answer, yes, I mean, some people will say that you can grow organoids of virtually everything now, and you probably can, but there are, I mean, the kidney is thought to be much harder, you know, other organs are easier. But yeah, over the last decade, there are a number of kind of improving protocols for development of these quite complex systems, really. So, you know, and even things like organ on a chip, where you can grow these organs with a kind of blood vessels and vasculature that you can test drugs. So I think technology is allowing us to develop a broad range of organoids with increasing complexity.
Thank you very much. I'm going to wrap up there and say thank you very much to our three speakers again. Excellent.